I think we should proceed.
Okay, great.
Yeah.
Thank you, Mason.
Sure.
Okay, well, good day, everyone. Welcome to the first CERC seminar of 2022. We're very happy that you're all joining us today. First off, I'd like to thank our sponsor, Active Motif, for helping us run this seminar series. Before we get started, another item is if you have any questions during the talks or after, please put them in the Q&A instead of the chat. That will help us manage the questions a bit better. Let's take it off. So it is my pleasure to introduce to you our first speaker of today, Frank Hu. Frank is a fourth-year PhD student under the supervision of Dr. Jacek Majewski, from who you'll be hearing later, as well as Dr. Michinori Saitao, who is at Kyoto University in Japan. And I heard that recently Frank was there as well for his research. So maybe we'll hear about that a little bit. So Frank's research focuses on integrating multiomics profiles of diverse model systems to obtain mechanistic insights into epigenomic dynamics of cancer as well as normal development. So today you'll be talking about nucleome dynamics in normal and stalled developments. So Frank, thank you so much for being here with us today. Whenever you're ready, please feel free to share your screen and start off.
Great. Great, so thank you for the kind introduction and giving me the opportunity to share our work describing and charting the 3D genome dynamics in both health and disease. And I will begin by discussing a project I undertook while in my co-supervisor, Dr. Michinori Saito's lab in Kyoto. So packed within each of them, every living organism's cells are strings of DNA molecules that serve as the fundamental building block of life. And within the encoded sequences that ultimately provide the blueprint for cellular function, and it is very critical to understand these genetic information. And so genomicists apply a variety of techniques to sort of profile and understand the variation in these critical sequences. And among the most popular approaches is to perform genome-wide association studies to sort of determine what genetic variants are predictive of traits and disease. While incredibly informative, rather than providing sort of actionable insights or like directly actionable insights, these studies often require further follow-up studies to obtain more mechanistic understanding. And one reason being that genes only make up 2% of our genome, with the rest being between genes, or what we call intergenic regions. And it was originally non-trivial to figure out what happens in these intergenic regions. And so as a result, there now exists substantial interest in interpreting genomic results through the lens of epigenomics, which studies the way in which DNA wraps around histones and how nanometer scale loosening or compaction of chromatin fiber can facilitate access to genes or intergenic regulatory elements, often involving chemical tags, such as post-translational modifications to these histone tails. In particular, chromatin immunoprecipitation techniques has been pivotal in characterizing the unique distributions of various histone modifications around various genomic regions, ranging from decondensed active regions to compacted silent regions. And altogether, these modifications jointly exert their influence in determining what happens at a local scale at various elements, say at promoters, enhancers, or broad domains. Yet an inherent limitation of ChIP-seq lies with the 1D view of our genome, whereas in reality, the genome adopts sort of non-trivial 3D folding patterns as our 2-meter-long DNA is compacted 1 million-fold to fit within our tiny cells. And this folding can be considered at distinct scales. For example, how enhancers loop around to activate genes, how chromatin can bunch up to bring transcription factors together, and so on. And to thus understand the nuclear architecture in our genome, what we call nucleome profiling, Hi-C is used to glue together pieces of DNA that are proximal to one another in 3D space. And by applying Hi-C to profile nucleomes, we can thus interrogate 3D genome organization at multiple scales, as mentioned before, all the way from chromosome territories down to intrachromosomal compartmentalization of chromatin into euchromatin or heterochromatin, as well as the organization of domains that are that critically forms the sort of a fundamental unit of transcription regulation, as enhancers are unlikely to reach across these boundaries to activate genes in other domains. And these domains are at their core formed by various architectural proteins, including boundary elements such as CTCF, and sort of these motor proteins that extrude chromatin called the cohesin complex. And so altogether, our genome, epigenome, and nucleome shape the sort of the global landscape that ultimately predicts and directs cell fate decisions, with say one potential readout being the transcriptome. And these decisions can range from one seen during normal development, such as a cell decide whether they want to say become a skin cell or a neuron, or when cells get sick during carcinogenesis. And thus a simultaneous understanding of multiple modalities are often necessary to truly grasp the mechanisms underlying a normal biological process or how disease arises. And so with our lab in Japan in particular, we're especially interested in vitro models of germline development. So here is an example of mouse spermatogenesis, where we can, in a dish starting from embryonic stem cells, ultimately derive fully functioning spermatozoa that is capable of providing viable and fertile offsprings. And so why we want to use this in vitro system is that, well, it's possible to isolate these same sort of germ cells and their progenitors in vivo. Their population is quite limited. And so it's very difficult to obtain sufficient material to apply most assays. And so for us, this project, we wanted to focus on the sort of the diploid development between the ICM stage and the spermatogonia stage, as this has been sort of less characterized than the preceding zygotic stages or the subsequent meiotic stages. And this critical period includes a number of key events, including the priming of stem cells, followed by the specification of primordial germ cells apart from the somatic phase, followed by the epigenetic reprogramming in which genome-wide DNA demethylation takes place, and ultimately the sexual dimorphic remethylation process as the cells take on a male-specific character. As there are already visible differences of chromatin organization under the microscope, we here set out to obtain a higher resolution description and sort of atlas of germline nucleome maturation. We immediately noticed that by simply looking at the chromosome scale high C maps, there appeared to be a gradual gain of distal interactions along germ cell development. As you see, the sort of the far parts of this map becomes redder and redder, indicating increased contacts. And simultaneously, so this is then sort of consistent with our observation of chromosomal decondensation that we are able to validate using microscopy and cytogenic techniques. When we further assess the interaction between euchromatic regions, sort of AA, or heterochromatic B regions, we saw that whereas euchromatic AA interactions gradually decreased during differentiation, heterochromatic BB compaction actually become heightened. And what's striking is that this observed both in cis, so intrachromosomally, and in interchromosomally. And sort of this high interchromosomal BB interaction came as a surprise, as many previous studies have sort of purported that interchromosomal contacts are usually related to sort of active transcriptional hubs, whereas here we find more of a predominance of interchromosomal heterochromatic aggregates in these germline stem cells, so spermatogonia. At the same time, the genome's overall characteristic also changes. In particular, we find a gradual euchromatization taking place across germ cell differentiation as the distribution of compartment scores experiences a gradual increase, and this sort of euchromatization trend stands in contrast to the repression found in at least 3 other lineages that we looked at, ranging from neuroinduction, B cell reprogramming, and cardiogenesis. And so we then sought to understand what specifically underlies these changes in global heterochromatic and euchromatic properties. Starting with silent regions by using H3K9 trimethylation ChIP-seq, we saw that these spermatogonial cells in particular saw the formation of very broad domains, often hundreds of kilobases in width. And they appear not as new domains, but largely arise as not as new domains, but largely arise as a consequence of expansion of pre-existing canine tribe peaks. And we further found that these domains overlap quite specific families of repetitive elements. Upon a closer look, we recovered a very specific association of this canine tribe spreading with line 1 elements, which is consistent with our understanding that pRNA-mediated repression of these transcribed young transposons being upregulated at this spermatogonia stage. And furthermore, these expanded canine tridomains not only lead to enhanced local compaction, but these domains also engage in distal aggregation, so between different domains. Finally, so this allows us to come to the conclusion that canine tri-mediated heterochromatic phase separation is the culprit of the elevated BB interaction we saw earlier. And this aggregation does not appear to take place anywhere in the cell, but specifically on the periphery. So for example, here, if we look at these GFP marked cells, which are germline stem cells, we see the enrichment of canine tri on the more or less on the periphery, whereas in the epiblast cells, we see that they do not show the same peripheral bias. So coming back to the euchromatic landscape, in particular the open chromatin regions that may have some regulatory function, we applied unsupervised clustering on the chromatin states of each open site in each cell type. And we robustly uncovered a number of distinct classes, such as enhancers, promoters, and CTCF binding sites. And this then allowed us to appreciate that in this day 4C7 stage, which corresponds to the global DNA demethylation, that there is a lot of these enhancers becoming active. And also in the following spermatogonia stage, there is a loss of these CTCF binding sites, which we will come back to soon. So in these day 4C7 PGCLCs, as we mentioned earlier, they're globally hypomethylated. And so we sort of expect things to turn on, such as enhancers. But actually, there is no sort of pervasive transcriptional upregulation. So what's keeping those enhancers from activating genes? And it turns out that there's actually fewer enhancer promoter loops. And the remaining enhancer promoter loops are actually closer in space. And when we took a look at sort of the domain organization, we're able to show that using multiple approaches, that this day 4C7, this hypomethylated stage, have the most TAD boundaries. And also using an independent approach, we were able to show that it has sort of the smallest boundaries. So sort of these are equivalent statements. Yet this elevated insulation is not maintained as we progress in the male fate, as these germline stem cells, these spermatogonial cells actually show a loss of CTCF binding, both in the whole cell lysate and specifically for the chromatin binding fraction. And so as an example of how this is impacting or influencing biological processes, let's take a look at this DDX4 locus, which is a key germline gene. And we find that there's a CTCF peak here insulating this gene, as shown by sort of this red stripe here. And it's not being like very much, it's not being super much like activated, as shown by this active transcription mark. However, when in GSCs, as the CTCF peak binding site is removed, we see that this DDX4 gene then makes a strong interaction with the downstream enhancer, resulting in the robust upregulation. And when we took this genome-wide by systematically identifying similar cases of concordant loop formation and upregulation, or loop dissociation and expression downregulation, specifically straddling these CTCF depleted sites, we found that the upregulated pathways specifically involve those we are known involvement in germ cell development, for example, pRNA, as mentioned before, or say male gamete generation and reproduction and so on. And so to further sort of confirm the biological relevance of these observations, we're able to use the artificially induced germline stem cell-like cell, so GSC-like cell type using suboptimal conditions. And the suboptimal conditions then leads to sort of a GSC LCs that have reduced or impaired spermatogenic potential. And so this then allows us to see, like what is causing this impairment as compared to the fully functional germline stem cells. And sort of at the global scale, we immediately saw that it's sort of like a intermediate compartmentalization state as compared to these precursor primordial germ cells versus the more fully functional GSCs, as we see the compartments being sort of more middling. And also coming back to CTCF insulation, we saw that GSCLCs having sort of these residual CTCF peaks that are not completely removed. And this again, so this is coming back to the DDX4 locus, this failure to remove CTCF results in a failure to completely upregulate, as we show here that the K36 tri is lower in GSCLCs. And globally, there is indeed reduced, there's indeed sort of more CTCF binding sites in this GSCLC cell type than this fully functional GSC cell type. And again, taking this genome-wide, we can confirm that the residual peak CTCF sites in GSCLCs are located near genes involved in important phenotypes, such as male, sort of delayed male fertility, providing further evidence of this the importance of CTCF. So as a summary of the results I presented today, we uncovered important functions of chromatin insulation in this hypomethylated stage. And we then say that this insulation must be removed for the subsequent activation of the spermatogenic fate, and failure to do so can then lead to reduced spermatogenesis activity. And so, of course, as I mentioned before, none of this is possible without the guidance offered by my lab members as well as collaborators who actually generated a huge amount of data, as well as help with the analysis. And finally, I will acknowledge the funding sources and thank you for your attention. And I'll be happy to answer any questions.
Great, thank you so much, Frank. That was a great talk. Very interesting. Let me open the Q&A. I think we have time for maybe a few questions. Oh, actually, I see that Marco, who unfortunately cannot use the Q&A because he's also a host, he has a question in the chat. Marco is wondering, what controls CTCF levels during cell state transitions?
So we did not specifically, like, say, look at the promoter of CTCF and sort of the global level of CTCF. So I'm unfortunately, I don't have a clear answer for that at the moment.
Okay, thank you. If anybody has questions, please put them in the Q&A. If we don't have time to go over them now, we can always do it later. I do also have a question. You earlier on in your talk, you were talking about how in the male germline, you see this euchromatization that you don't see in other cell types. Do you have any hypothesis as to why that might be the case in the male germ cell, but not in other types of cells?
Right. So I think there's been previous reports of saying us in spermatogonia, as they sort of prepare for meiosis, they really need to be in fully active mode, and so they're preparing to produce lots of proteins. And so I think it's sort of it's perhaps not unexpected that there's a gradual sort of ramping up of transcriptional activity per se.
I see, that makes sense. We have a question from Divya Singhal. Great talk, Frank. Just curious to know how those dynamic chromatin maps were achieved.
The dynamic, I was saying on the high C maps, or so for example, I could talk a bit more like how high C is done. So essentially, I'm not sure if they meant high C or some other thing. For example, in high C, we just ligate pieces of the cis regulatory map. Yes. So here, for example, we have a for each open site in a cell type, a sort of a row in a matrix, and then the columns or the features of the matrix are different epigenetic modifications, say K4 tri, K27 tri, K36 di, and so on. So we have a square matrix or a rectangular matrix, and this is just we can apply, say, dimension reduction or unsupervised clustering to that matrix. It's very much sort of taking cues from single cell sort of clustering and so on.
Thank you. There's a question from William Pastor. I'll take this as the last question before we have to move on. He's asking, is there a pattern to which CTCF sites remain versus are lost during development? anything in terms of motif chromatin that replaces it, etc.
Right, so I didn't get a chance to mention it, but like in terms of the chromatin state, we found that actually there's some loss of CTCF is not happening in heterochromatin, it's happening in euchromatin regions, and they tend to have high K36 methylation. So we think sort of like as the genome is remethylating from the fully demethylated state, this K36 methylation and actually the DNA methylation, the novel DNA methylation that they recruit is sort of evicting these CTCF.
I see. I see there's another question from Matt, but maybe I think you have access to the Q&A, so maybe you could answer him in there. That would be great. Okay. Then we will move on now. Let me organize my screens. Okay, so next, it is my real pleasure to introduce Dr. Jacek Majewski. Jacek is a professor of human genetics at McGill University. He received a PhD in evolutionary biology from Wesleyan University, after which he joined the lab of Dr. Jörg Ott at the Rockefeller University for his postdoc in statistical genetics. Jacek's lab focuses at understanding basic biology, hereditary diseases, and cancer. And an example of his recent seminal work is the work on mutations in histone genes like K27M. Today, he will be talking to us about nucleosome dynamics in disease, and I think he's going to talk about pediatric glioma, if I saw that correctly earlier. I'm really looking forward to your talk. Please take the stage.
Is my screen visible?
Yes.
Okay, great. So thank you very much for the invitation. I will indeed be talking about glioma, and in a way, it's a continuation of Frank's talk. Now we're going from nucleome dynamics in normal development to nucleome dynamics in disease. But before I get there, I guess I will have to give a little bit of background about the earlier work on glioma. So the pediatric high-grade glioma is a very deadly disease with 5-year survival being very rare. And because it is so deadly and because adult glioma is more common than pediatric glioma, this was one of the first cancers that was tackled by the Cancer Genome Atlas Consortium. And they found some very interesting things as far as the genetic mutations that cause gliomas. And they found molecular classifications that were quite meaningful. And that was about the year 2010. And this is when I met Nada Jabbar. And Nada had the foresight that she often exhibits in her research. She came to me saying something that pediatricians often say. She's a pediatric oncologist, that children are not just small adults. But she had good reasons for saying this in this case, because the tumors just, despite the fact that they look histologically very similar, they had some very different characteristics. So she convinced me, our group, to do some exome sequencing on the tumors that she had available. And we did the analysis, and I had the privilege of being, one of the first people to discover some, still to me, incredible results, something very unexpected. And what we detected in this patient cohort were mutations in histone genes themselves. And at the time, I was, an avid geneticist, but I was not so knowledgeable about epigenetics. So it took me a while to fully understand the significance of this. That it's the histone genes, the very building block of chromatin, and the very specific residues that are very important in chromatin modifications that were being mutated and were somehow responsible for pediatric gliomas. And we found 2 recurrent and very prominent mutations, and I will concentrate today on this one, the histone 3 lysine 27 to methionine mutation. So we found it in 2012, and later, as we were studying and doing more sequencing of the patients, we realized that this mutation usually pretty much always co-occurs with more typical cancer oncogenic mutations such as P53 or EGFR or ACVR1. And there are very specific associations that are characteristics of the age and the geographic location within the brain. So it enforced in us this idea that pediatric glioma is a developmental disease and that these mutations have to occur at a very specific point of development and a very specific cell type in order to, well, do whatever it is that they're doing. And the idea is that that became more prominent as we were thinking about this is that there must be a developmental blockade that underlies this pediatric glioma oncogenesis, and it's this histone mutation that somehow must be causing this blockade. And we later found that this H3K27N mutation is either the earliest or in some cases co-occurs immediately with other mutations. So it's the driver oncogenic mutation. And through research mainly from Claudia Kleinman's lab, we were able to pinpoint the cell of origin as glial committed neural progenitor. So, we already, at this point, we keep learning more and more about the pathogenesis, but let me go back a little bit more to the mutation itself. So I don't have to get too much into this, but obviously chromatin epigenome is very important. Lysine 27 is very important. We know that methylation is generally associated with facultative heterochromatin, especially trimethylation. Acetylation is associated with opening up, loosening of chromatin promoters and enhancers. So these two modifications have very different significances. And the big question is, so now we're mutating these very important residues. What exactly is happening? What are the immediate downstream effects? And very soon as we were thinking about this, we realized that there are people who know much more about epigenetics than we do. And we started a collaboration with David Alice at the Rockefeller University in New York. And David, and at that time, his postdoc Peter Lewis, did some very early work to show that if you look at histone mutant diffuse intrinsic point in gliomas, and in this specific tumor, the histones 3 K27M mutations, they're present in 95% of the patients. It's very rare that you don't have this mutation. But if you compare the mutant tumors with the wild type, you find that two things are obvious, is that lysine 27 trimethylation decreases, which we expected because we are now changing the lysine to methionine so it cannot be methylated. But it was very unexpected to see that lysine 27 acetylation actually was elevated. And that led to the discovery that this lysine 27 to methionine mutation was not actually just acting locally, but it was a global inhibitor of the polycomp repressive complex 2, PRC2. And most likely what is happening is that this methionine at this position has a higher affinity for PRC2, or PRC2 has higher affinity for this methionine than the lysine. And when PRC2 stably binds to chromatin, which is necessary for the position of lysine 27 trimethylation, it binds way too strongly and either is not released, or there's another theory that when it's released, it's sort of misshapen or unable to further do its job. And it's not fully clear yet, but the result is that it's even a very small fraction of this mutant histone can totally inhibit or very strongly inhibit PRC2. So having learned that, we started doing some work in cell lines, and this work was done mostly by Ashot, a postdoc in Nada's lab, Simon, a student in my lab, and Brian at that time, beginning PhD student also in Nada's lab. And we started working with patient-derived cell lines, and we could do a number of manipulation with them. So we could take wild-type glioma cell lines and mutate them into the lysine 27 to methionine, mutate the histone to methionine. We could take the mutant histone and revert it to the wild type. We could overexpress a bunch of PRC2 components and see what happens. And after all this, we could do a lot of genomics and try to figure out what the genome-wide effects of these mutations or lack of those mutations or manipulations of other epigenetic components are. So here's what we found that was really cool. So if you look at the top track, it's the wild-type glioma cell line, and the blue track displays K27 trimethylation, and normally in those cells, K27ME3 is present in these large heterochromatic K27ME3 domains, and they can be hundreds of thousands or megabases long. When we introduce the mutation, we see that it's not just that the levels of K27ME3 decrease, but they decrease within these domains and only remain at very specific regions, like here, if you follow my pointer. And those regions correspond to SUS12 or PRC2 enriched region. And these are CPG islands that are usually enhancers of promoters of PRC2 target developmentally important genes, inactive genes, ones that are not expressed. So pretty much the only place where we're retaining K27 methylation is at those nucleation sites for PRC2. And it's gone from most of the rest of the genome. And we see the same effect when we look at mutant cell lines that normally have the K27M mutation after we remove them. So at the top here in red, we have the kind of the peaky distribution of K27ME3 in those mutants. When we remove the mutation, K27ME3 is able to spread into those domains. And the same thing happens in other cell lines that we look at. So here's the model that we came up with and that I still like quite a lot. So in normal cells, you have these inactive promoters, CPG islands of PRC2 target genes, and PRC2 will bind to it. It will deposit K27ME3 in this kind of peak around it. And then as the cell development progresses and the cell ages, PRC2 will diffuse outwards from there and will deposit K27ME3 in these broad domains. Can you hold on a sec? Just give me a second. My son is playing piano, and I will ask him to maybe hold off for 15 minutes.
Of course.
So we have these in a normal cell, these broad domains, and what happens in the K27M mutant is that PRC2 still binds to its nucleation site. And it deposits this peak around the nucleation site. But then when it starts diffusing out, it will encounter the K27M mutation. It will become deactivated. As a result, all we have is this kind of scraggly peak around where the domain should be. So we don't have this domain. And here's the comparison of the normal and the mutant. And within this region that normally would be occupied by K27ME3, now things can happen. So some promoters within this region can be silenced by DNA methylation. Other promoters will acquire K4ME3, or maybe they already were bivalent and have K4ME3, and they will start being re-expressed. And then these regions also become acetylated. It's a whole, other story of what happens in those regions. And we have published quite a bit on that. Others have also. I will not talk about it today. I just want to mention that these regions that are now devoid of K27ME3 are sort of de-repressed and primed for transcription. And we do see that using RNA sequencing. And generally what we see is that here we divided gene expression into deciles, and it's really the lowly expressed genes or genes that were silenced that become very lowly expressed. So it's not like a massive upregulation of genes. It's more of a transcriptional noise of genes that were repressed by these domains that now become, somewhat active. And to sort of re-emphasize this and to move on, here's another screenshot, and we have this peak of nucleation site of K20 of PRC2, K27ME3. In the normal cell, we have this large domain, or the wild type cell without the mutation. We will have the creation of this large K27ME3 domain. It does not happen in the mutant cell. As a result, in the mutant cell, within these depleted domains, we have weak de-repression of genes that normally should be repressed. So this is one thing, and from now on, we're going into a new territory. We had shown this quite a long time ago. So the question we want to ask here is, if you look at these peaks remain at the promoters, the nucleation sites, and as far as we can tell, these peaks don't really decrease. They're of comparable size in the mutant and wild type normal conditions. So what happens under the peaks? This is sort of the title of this continuation, this new part of the talk. And well, the simple answer is that those peaks maintain silencing. So genes that are PRC2 targets, strong CGIs that recruit PRC2, they are not de-repressed by this K27M mutation. And looking into this in more detail, this is the work of Frank, who you just heard, Brian, student in Nada's lab, and Haifeng Chen, who's a postdoc in my lab. Brian did most of the wet lab work. Frank and Haifeng did most of the analysis. So what happens under the peaks? Well, now we have to go into a bit more into interactions between different epigenetic modifications. So we're not just looking at K27ME3 anymore and the silencing within the domains. We are looking how PRC2 and PRC1, another component of the polycomb or another component, I guess, of the polycomb repressive complex, how do they interact? I won't go into detail about different flavors of PRC1 and PRC2. I'll just mention that canonical PRC1, which has these specific CBX and PHC subunits, can read or recognize and bind to, so using the chromodomain, to K27ME3, which is the product of PRC2. So this is sort of a key. The catalytic product of PRC1 is H2AK119 ubiquitination. The catalytic product of PRC2 is K27ME3, and the specific flavor of PRC1, canonical PRC1, is able to read the product of PRC2, right? So they're interdependent. And it has been shown now through several studies that PRC1, through this PHC subcomponent, contributes to the formation of something called polycomb bodies. So these are these nucleation sites of PRC that come together, and they form this very tightly, in the term of 3D architecture, these tight loops that are silencing loops, right? So they kind of compact things to the extent that whatever is at the edges of these regions that come together become stably silenced. And it has been shown in developmental system that during development, these loops kind of dissolve, and it allows expression of developmentally important genes at the right time. So now in our system, we have the substrate, which is K27ME3, which in order to produce these polycomb bodies is being recognized and read by canonical PRC1, which leads to the compaction. And if you think about it, histone And so histone and histone methylation is very, very abundant in the genome, right? Histones are the most abundant proteins. Its methylation can be 10, 10%, 20%, 30%, depending on the cell type, and this is the trimethylation. So there's a lot of K27ME3 in a normal cell, in some cell types, especially as they mature. On the other hand, there's relatively little PRC1. So the recruitment of PRC1 will be very dependent. It will be, PRC1 is like the limiting factor in the creation of these polycomb bodies. So the recruitment of PRC1 will be very dependent on the distribution of K27ME3. And well, what is the distribution in the systems that we're looking at? Glioma, pediatric gliomas, and also ependymomas, which is dependent on this EZIP gene, which kind of acts a little bit like K27M. It also suppresses PRC2 activity. So in the presence of these in the oncogene K27M or EZIP, we have what we call it, well, we refer to as a peaky distribution of K27ME3. And when we get rid of this mutation, we have a more broad distribution. And the same thing happens during development. There are stages with very pointed, focused distribution of K27ME3, and there are stages with a very diffuse distribution. And this developmental part we're doing in collaboration with Chao Lu at Columbia University. I will not discuss it much today. I will just focus on the pediatric tumor cases. So what we're seeing in developing brain, generally during development of the brain, is that K27ME3 in early stages is confined, like it is in pediatric glioma. And then as the tissue cells type neurons develop, the K27ME3 distribution invariably becomes more diffuse. And what we're seeing at the level of chromatin compaction and these polycomb loops is that in the K27M mutation, where we have very pointy distribution of K27ME3, we have very strong interactions, these dots that connect the PRC binding sites are very strong. We don't see this when we knock out the mutation, right? This loop here, this interaction between different K27ME3 peaks becomes much more diffuse. This is an example of a Hox cluster, which is one of the strongest examples in the genome. There's many other examples here, CDKN2A, which is a well-known oncogene, and similarly, it has a very strong loop in the presence. Its promoter has a very strong loops in the presence of K27M mutation, but when we remove the mutation and the distribution of K27ME3 becomes diffuse, this loop pretty much disappears. And we can average this genome-wide, so we can take all these PRC target CGIs at this point and combine the information from the genome-wide. And what we find is invariably in the condition where we have peaky K27ME3, we have on average these very strong polycomb repressive loops. When we remove the mutation, those loops dissolve. And this is true across the board, right? So this is dependent on K27ME3, and we think this is mediated by canonical PRC1. Well, is it? We've done some more ChIP-seq. We've provided different components of PRC1, specifically CBX2, which only exists in canonical PRC1. And we can see that indeed the distribution of CBX2 is very punctate in the presence of K27M, and it becomes diffuse when we knock out the mutation. We can also, this is not seen for H2A ubiquitination, which is the catalytic product of PRC1. So we're seeing more and more evidence that it actually is canonical PRC1 that is responsible for these for these strong repressive loops in the presence of K27M mutation. So this is another one of those plots that Frank made, and I guess maybe it'll be easier to explain here. We actually see the different epigenetic markers that he uses to make those clusters. This looks like single cell sequencing, but it is not. Every dot here represents a promoter or an enhancer, a CPG island, and they are clustered using these different epigenetic measures that we make. And Frank was able to take all of these cells and subdivide, or the CGIs, subdivide them into active, which are active, other, which are usually not active, PRC2 targets, so these are CGIs that have K27ME3, and canonical PRC1 targets, so those are the ones that have usually both K27ME3 and CBX2. So I'll skip this one. So here's what we think is happening and sort of the hypothesis that we're pursuing further. So in the presence of the mutation, we have this focus distribution of K27ME3. This leads to a focus distribution of canonical PRC1, and this leads to these strong repressive loops between PRC target promoters. When we remove the mutation, K27ME3 spreads out. It titrates away canonical PRC1. Now these CBX2 peaks become less prominent because PRC1 goes elsewhere in the genome, wherever it can find K27ME3, and these loops sort of dissolve. And when here we look at the associated gene expression, it fits in the presence of the mutation, PRMD13, the gene at the base of this loop is lowly expressed. When we knock out the mutation, the loop dissolves and the gene becomes more highly expressed. So we tried doing this on the genome-wide basis, and we compared expression of all genes within those 4 clusters that we defined earlier. In the presence of the mutation, K27M, compared to when we knock out the mutation, and the active genes remain active, the other inactive genes remain inactive. The PRC2 target genes actually go down when we don't knock out the mutation, which means they become repressed, which is consistent with what we've seen before. So these are the genes under these broad domains of K27 methylation. But the PRC1, canonical PRC1 target genes actually don't seem to change. So that was a bit disappointing until this experiment was done in stem cell media. But then Brian tried differentiating those cells, and we saw something quite different. So when we try to differentiate the cells more towards astrocytic lineages, we see that specifically those PRC1 target promoter genes now become de-repressed, and when these loops dissolve, when K27M mutation is gone, when we don't have the peaky distribution of K27ME3, when we don't have focused PRC1, now these target promoters are able to be expressed. Again, this is low-level expression, but they do become de-repressed. And so this was all genomics. We can look at this in more functional level, and we see that in the presence of the K27M mutation, we can take the glioma cells and we can try to differentiate them. They don't want to differentiate. When we knock out the mutation and we do the same experiment, they now start expressing GFAP, the astrocytic lineage marker. So let's see, the mutation is preventing differentiation. And probably even maybe more importantly or more prominently, if we remove the mutation, so we can take these tumor cell lines and we can inject them into mouse to propagate as xenografts. They do grow and the mice die. If we remove the mutation, the cells do grow, but they don't really form tumors. So when we look at the xenografts in the presence of the mutation, we see very strong tumors. In the absence, we can find the cells, but they are not forming a tumor. And we can look at the expression of genes within those cells. And I don't have time to get into detail, but we do see by gene expression that in the presence of the mutation, we are not differentiating towards, we see more of the undifferentiated cell line markers. In the absence of the mutation, when we knock it out, these cells seem to be differentiating better towards, well, what we think they should be differentiating to if the mutation did not happen. So one last thing I want to end on is, well, we think all this is mediated by this focus distribution of K27ME3 that is then mediated by focused PRC1 and excessive loop formation at these polycomb bodies. Well, can we inhibit PRC1? Can we prevent it from binding to K27ME3? And we can using this allosteric inhibitor. And let's look at this again, what happens when we use the inhibitor. This is again the differentiation media. And if you remember from previously, nothing much changes except for these canonical PRC1 target genes that become upregulated in the absence of the mutation in the knockout, right? So now we're doing the same thing. We're treating the K27, we're not removing the mutation, we're treating the mutant cells with inhibitor. And the active genes don't change, the inactive genes don't change, PRC2 targets don't change, but the canonical PRC1 targets actually are upregulated, which is in line with the idea that that we're targeting PRC1, and PRC1 was responsible for repressing those genes. They don't become as upregulated as in the absence of the mutation, but they go in the right direction. And we can see this also in the differentiation series. So when we look at the K27M cells and we differentiate them, well, they don't want to differentiate. When we remove the mutation, they now express GFAP, they differentiate more towards the astrocytic lineage. And when we treat the mutant cells with the PRC1 inhibitor, they are also now easier to differentiate towards astrocyte. So this is really exciting. This is really, it points us towards some therapeutic vulnerability that we can explore. So we have this pediatric cancer that is stalled in some developmental stage that it should not be in, and it's proliferating instead of differentiating. Well, we can try to kill it. So far, we haven't been able to. We can try to push it in some other direction. EZH2 inhibitors so far have not worked. Other epigenetic drugs have not either. We can try to, make it differentiate into something else. But now we think we have a way to help it differentiate it towards what it should be differentiating into, and hopefully reduce the proliferation of the tumor. Well, obviously, the next step will be to try this inhibitor in mice on the xenografts, and hopefully, if you invite Brian or Nada next time, they might tell you about those experiments. But I want to end here on a optimistic note that we have understood this tumor at this point pretty well, and we think we have tools to start treating it. Okay, so summary, 10 years of progress. We think that pediatric glioblastoma is a developmental disease, and specifically a disease of stalled development. The primary effect of this K27 mutation is inhibition of PRC2 and this focus distribution and lower levels of K27ME3. But a cascade of all different epigenetic rearrangements follows. I have not talked about all of those or most of those rearrangements. I really just talk about the relationship with PRC1. And two things happen. It's not simple. So we have some genes that are de-repressed by the presence of K27M mutation and lack of formation of those large domains. At the same time, we have some genes under those peaks that are excessively repressed, and they are not released from developmental arrest. So this results in blockade. And well, we think we have a way to release the cells from this blockade. So I will stop here. And of course, just acknowledge the funding and, well, not all the collaborators, because this has been a really large project here. I'm just listing people who have been directly contributed most to what I presented today. And it's my lab, people from Nada's lab, Claudia Kleiman, Chao Lu from Colombia, and Ben Garcia, who now is in St. Louis. So thank you very much. And yeah.
Thank you so much, Jacek, for a great talk. If anybody has questions, you can either put them in the Q&A or you can raise your hand if you prefer to ask them in person. So I think let me open up the Q&A. Oh, there is already a question there from Patrick Laurette. He's asking, does this K27M mutation impair differentiation into other lineages?
That is a really cool question. My answer will be weak, but overall the answer is yes. These mutations are found in some other tumors, in okay, I know they're found in leukemias, and they have, a similar effect of impairing differentiation. So, my feeling is that since in most systems, as development differentiation progress, you have this property of, taking lysine 27 methylation and spreading it further throughout the genome to form these domains. And this seems to be associated, and I believe it's instrumentative to normal development. If you prevent this from happening, my prediction is that in most cells, this really will prevent development. Why do we only see these mutations, or mostly see these mutations in gliomas? Well, that's kind of another fascinating thing to me, that most cancer mutations are so specific. They really have to occur at the right tissue, at the right time, at the right cell type. Otherwise, they can just probably kill the cells. So in some cases, they will be oncogenic. In some cases, they will probably be detrimental to the survival of the cell. But yeah, I guess my answer is, in the systems I know of, this mutation prevents differentiation, and my prediction would be, in most cases, it would probably be detrimental to differentiation.
Thank you. Then there's a question from Jamie Kramer. It's a large question. I'll try to maybe break it up. So he says, very interesting talk, Jacek. Is the K27M mutation in the variant histone gene H3.3 and not the canonical histone H3 genes?
That's a really exciting question. And going back to the picture I flashed in the beginning, mutations the mutations occur, these mutations occur in both histones. They occur in 3.3, 3.1, and actually also 3.2, which is just slightly different from 3.1. But yet they, it depends where in the brain these tumors are, will dictate which histone is mutated. And usually the older patients have H3.3 mutations, the younger patient in the brainstem has H3.1 mutation. The H3.3 usually co-occurs with P53, H3.1 more often co-occurs with ACVR1 mutations. So there's something there to do with cell cycle. There's something there probably to do with just the levels of expression of the gene. I know that Nada's lab is working on this very, very hard, and I think they have a manuscript that they will submit pretty soon, if they have not yet already. But I don't know all the details of what they found out yet. I just know it's absolutely fascinating that this can occur in different histones, but not in the same cell type and not with the same partner mutations.
Thank you. Before I will continue asking the next questions, but since it is 10, just in case people have to leave right now, I just wanted to mention that our next seminar is going to be on February 25th by Dr. McGill and one of his trainees. Okay, so I'm going back to the next part of the question from Jamie. So going back, if so, does the mutation affect anything about the incorporation of the variant histone H3.3 in the genome? And then have you compared your epigenomics data in the mutant cell lines to the distribution of H3.3 in the genome versus canonical H3, which presumably is present and available to be methylated?
Right. So, that's another fascinating question. So the canonical H3.1 is sort of evenly spread throughout the genome. It does not have any known preference that we can see. The variant histone H3.3 is mostly located in active regions of the genome, so gene bodies, promoters, genes where histone turnover happens. So, the basic mechanism that I have in my mind is that during replication, you put, there's a burst of histone incorporation. It's mostly H3.1. And then as the cell kind of goes through its stages, in regions where this histone turnover is being replaced with H3.3. So H3.3 has this specific distribution throughout the genome, and we thought, it still may to some extent be related to the to its epigenetic consequence. But as far as I know, we are not able to see any difference between wild type H3.3 and K27 and mutant H3.3 as far as where they're incorporated. They seem to go to the same places. And since the outcome or the effect of the mutation is global, it's not a localized inhibition. It inhibits PRC2, dominantly. My feeling is that the distribution of H3.3 is not so important, but it may well be. This is just kind of my intuition, but I don't know.
Thank you. Then there's a question by Matt. He says, thanks, Jacek. Do you think the observed looping is actually required for silencing of relevant canonical PRC1 targets that you describe versus in cis effects of the presence of canonical PRC1 and K27 dry over affected CGI promoters?
Yeah, that's a good question, and that's probably something that we would like to follow in the future. Right now, our hypothesis is that this looping, the stable association, or the strong association, and the creation of these polycomb bodies actually contributes to the silencing. There probably are some experiments we can do in the future to dissolve the loops without disturbing PRC1 and K27 ME3 levels at those regions. That would be interesting. For now, we don't know, but our hypothesis is that those loops are actually causative.
Okay, great. Thank you. Then there was a question from Marco actually in the chat. Do you think what you observed about CBX2 is also applicable to tumors with EZIP overexpression or / mutations?
Yes, we now I don't. Most of the functional work we've done on gliomas, but we do think that in the posterior foci group A ependymomas that express EZIP, well, the situation is very similar. We see those focused regions of K27 ME3 deposition around promoters. I don't remember if we actually profiled PRC1. So, unfortunately, I don't remember the data that well, but it is our hypothesis that the same thing happens in ependymomas.
Okay. Thank you. I think that were all the questions for now. So with that, I would like to thank our speakers, Jacek and Frank. Thank you so much for 2 amazing talks. And thank you everyone who was here today. We hope to see you all back on the 25th when Mathieu Lepiane and one of the trainees from his lab will be giving a talk. So thank you all and have a great day.